All right, so our next speaker is Dr. Jonathan Carlson, a senior researcher from Microsoft Research, a senior scientist. He started his career in computational biology with an undergrad degree in biology and computer science from Dartmouth in 2003. There he acquired a love for virology, which provided the focus of his thesis work as he pursued his PhD just south of the border at University of Washington. And during his PhD, he was co-supervised by a research scientist at Microsoft Research. So in 2009, he had a pretty seamless transition to be essentially a scientist for them in 2009. And since then, he's been a prolific researcher in the field of virology. And today, he's going to give us a taste of Project Premonition. And with that, let's give a nice warm welcome to Dr. Carlson.
Sorry, I'll try not to roam around so that the recording works. Well, thank you. Thanks to Will for inviting me up here. I've been looking for an excuse to come up to Vancouver since I moved back to Seattle a couple of years ago, and this is an awesome opportunity, and even sweetens the deal with beer and pizza afterwards. So I'll try and keep this somewhat short. So most of my work has been, I'd say 98.5% of my work has been on HIV. So what I'm going to talk about today has nothing to do with most of my work. It's going to be a departure for me. This is a project that we started about a year ago, and I'm getting more and more excited about. But Will said he wanted me to focus on an ongoing work instead of past work, so that's very much what this is. Yeah, I look forward to your feedback and discussion over beer and pizza afterwards. So what is Project Premonition? First, let me introduce the team. So this is a truly public-private partnership. There's a number of PIs on this. This was really set up by Ethan Jackson, who is a programming language expert at Microsoft Research. And he really pulled together this team of people from several centers. I'll point out in particular Jim Pippis, who's our virologist, Doug Norris, who's our entomologist. Anna is also an entomologist. We have a number of robotics and autonomous systems experts. And Ethan spent about a year or two convincing us all that this was an interesting idea that we should pursue. And then once he convinced us all that we should pursue this, we had a funding problem, so we went to IARPA and really put together a joint project between Microsoft Research and IARPA. And what we got was a one-year seedling. So this was 1 year to sort of figure out what was going on. And in the words of IARPA, our goal in this year was to move them from disbelief to doubt. So if at the end of this talk you have serious doubts, I will consider that a win. So the idea of Project Premonition is to look at the issue of disease epidemics. We all know that that emerging infectious diseases is a major problem. We keep getting hit by re-emerging diseases, new diseases, and whatnot. And the question really, at some level, was can we take advances in robotics, machine learning, sequencing, et cetera, and approach this from a technological perspective? And the key insight that Ethan had was that we can use mosquitoes as our sampling device. So you could imagine trying to build drones to go around and sample blood from animals and people, but that has some ethical issues and some technical issues. But it turns out that mosquitoes are really good at this, frighteningly good at this, in fact. And so if we could scope our problem not to sampling blood in the environment, but to sampling mosquitoes at scale, then that becomes a much more tractable problem. So the big idea is basically that most, if not all, problematic viruses have some animal host reservoir, but the mosquitoes are sampling the blood for us. So our goal is to design some traps that can actually catch these things at scale, and I will spend a few minutes talking about the traps. That's our B2 trap. We've still got a ways to go with that, but I will show you where we are there. Eventually, we want these traps to be deployed autonomously via drones. There's a ways to go with that, but of all the parts of this project, this is the one that we could probably have the most confidence will be solved. There's a lot of people working on autonomous drone flight, so whether we solve it or not, somebody will. And even before we solve that, you can imagine using drones as a way to sort of survey areas and identify mosquito hotspots. Once you get the mosquitoes, you can sequence them, and then the idea is to try and figure out what the mosquitoes ate in terms of host, and what viruses were in what they ate. And once you start doing that at scale, you can start putting together maps in terms of where viruses are. So let me first talk quickly about the traps. So we started this about a year ago. I will tell you up front, I had nothing to do with these traps. I think they're super cool, so I'm going to spend a couple minutes talking about them, but I'm going to spend most of the talk really on the metagenomics. So here's the state of the art for the traps. These were designed, I don't know, 20s or 30s at some point, and they really haven't changed much except igloos changed color, I think. So basically what you have is you have an igloo full of dry ice, which is your carbon dioxide source. The carbon dioxide attracts the mosquitoes. There's a little fan under here that as the mosquitoes get close, they get sucked into the trap. And it's not in the picture, but this whole thing is powered by a car battery. So you carry this thing out, you drop it, you hang it somewhere, and you come back 18 hours later, and you get something that looks like this. So then you have a grad student sit here and pull out the mosquitoes from all the not mosquitoes. So it depends on where you go. I'm told this is typically about 95% non-mosquitoes. We've been doing a lot of our partnerships with the city of Houston. They have a lot of mosquitoes there, so they actually get 95% mosquitoes, but that's a good reason maybe not to go to Houston. So what did our hardware team do? They said, this is an obvious place where we can innovate with technology. We have cheap microprocessors, we have cheap sensors. You can, from prototyping, you can 3D print these things. So here's what they came up with. I got one of these traps down here. Feel free to look at it afterwards. But the key part of this design was these individual cells. And the idea was, the big problem here is you come out, you set a trap, you catch something, it's probably not what you're looking for, and then you have to go through and figure out what's there. So what if we could only capture mosquitoes? One of the teams on this project from UC Riverside had developed a laser-based system to speciate mosquitoes based off the sounds of their wings. So the hardware team figured out how to move that from lasers to LED lights, which are much cheaper and lower power. And basically what we have are these little cells, these individual little cells right here. Each one of these is one of these squares, and what you have is a little LED light. The current design is a little different from this. You basically have a mirror, so the beam bounces back and forth, and then a little cheap CCD sensor. Those two things together are, at scale, a few cents, so these things are super cheap. And when a bug comes in and checks things out, it flies in here, it triggers a sensor, and you get a burst of light. This is all in the infrared spectrum, so the bugs can't see it. And when you get that, you can look at the wing beat frequency and classify whether you got a mosquito, and even, as I will show you, what type of mosquito you got. And of course, as long as we're building computers, you can have all sorts of interesting sensors, temperature, humidity, ambient light, etc. The whole thing is powered by a cell phone battery and communicates via Bluetooth in the current iteration. So what can you do with this? Well, here's a video in the insectary. You can see the mosquitoes here flying around. They probe. We can actually see the UV light here with the UV camera. But then as the mosquitoes fly in here, it triggers the... Am I going to go back in? Maybe? Maybe not. There we go. It triggers the sensor and closes the door. So this works splendidly in an insectary down UC Riverside. The next step was to try it in the field. So we partnered with Harris County in Houston, Texas, to see if this would work in the field. I'm going to keep this short, but in short, you can pretty easily, just from the wing beat frequency, distinguish not just mosquitoes from everything else, but even the different genus of mosquitoes. So Culex is the West Nile vector, feeds mostly on birds, but also on humans. And then Aedes is the Zika vector and carries all sorts of other nasty things. So this is just looking at wing beat frequency. You can see you can get a really nice separation from that alone. And once you can do that, now you can start asking interesting questions like, when are these things feeding? We set this trap up in the morning, we come back the next day. Typically you'd have no idea when you caught the mosquitoes, but here we know exactly the point in time when they encountered the trap. So you can make plots like this. This was after about 2 weeks of data collection with, I think, 10 traps, 20 traps in Houston. And what you see is immediately you can see the circadian rhythms, the feeding patterns of these two different genus of mosquitoes. So in purple here we have the Culex. We can see they're night feeders. As soon as sunrise hits, they stop eating and they pop up again when night falls. Whereas Aedes sleeps all night and then has these bursts throughout the day and really likes to feed right at sunset. So we happen to already know this for these mosquitoes in an insectary, but it was pretty amazing to be able to see this within 2 weeks of capture. And when we think about the big picture, there's 36,000 species of mosquito, and we understand those that we can raise in the insectary, but the vast majority we can't. So all of a sudden now we can start getting all sorts of really fine-grained details about these mosquitoes. And of course, we don't know what these peaks are. They may just be noise in the data, but we can start asking interesting questions, right? Because we not only know the time, we know the humidity, the temperature, the ambient light, all sorts of interesting questions. So from the entomological standpoint, this becomes an interesting starting point to really start to learn about the foraging behaviors of these mosquitoes. So next steps for this, basically we need to make the trap smaller and lighter, reduce costs, etc. I will say that the hardware team thinks that at scale these costs are roughly going to be the same cost as existing traps, which sort of goes to the pace of technology and what happens when you have a field that forgets to innovate in their space. And you know, we're working on building this all into cloud analytics. All right, so the main part of this talk is not to talk about traps, except I thought they're cool, so I want to talk about that a little bit. Mostly what I want to talk about is the metagenomics, which is what I've been focusing on. And when we think about the metagenomics problem, really what we're talking about is taking a mosquito here and trying to understand what it is. Now, typical metagenomics problems, you basically say, okay, here's a sample from human. Let's subtract out human and see what else is left. In this case, yeah, we can subtract out the mosquito, but what's left could be human, rat, mouse, deer. Goodness knows what else could be in there. So we've really cast our problem statement larger. We've set our problem statement to be, given a set of genetic sequences and zero background information, identify the set of source species in their relative proportions. So we're not there yet. I think we've made some good progress, but this is what I want to talk about as what our approach is and where we're at for that. And my hope is that this will be applicable beyond just the mosquitoes. So here's the current state of the art. I would say this is SERPy, which is becoming more and more widely used for virus identification, certainly within humans. And basically the pipeline is you subtract out host. Depending on where you get your sample from, you'll throw out 25 to 99% of your reads by subtracting out the host. You'll then align to some reference, viral, bacterial, whatever you're looking for. And then you'll basically count the results and make some pretty reports. There's some problems to this. What if the host is poorly characterized? So we do have mosquito genomes, but we only have a few of them, and they're not very well put together. They've got lots of contamination issues. And this is actually, we've seen this problem in other places with ticks, with birds. Oftentimes when you're in the environment, you don't really have a well-characterized host. The references tend to ignore most of the actual references. So RefSeq is really big. In our case, we really want to look at RefSeq because we don't know what the mosquito bit. We want to know if it was a mouse or a dog or a deer or whatever it was. And once you expand the reference, counting becomes a huge problem because a lot of these reads are very similar, right? So we are quite similar to monkeys, mice. You'd be surprised how similar we are to mosquito, actually. So simply counting becomes a huge problem at that point. So our approach is to drop the subtraction, to really align to everything we can get our hands on. And then rather than simply counting the results, use probabilistic models to assign reads to taxa, basically by sharing information across the reads. So that's what I'm going to talk about mostly today. There's some other things that people typically do, de novo assembly, coverage maps and whatnot. We're not approaching that right now. I think we'll get there eventually. Mostly our plan is to take existing solutions and plug that into our pipeline, but I'm not really going to talk about that. All right, so the reference databases. Currently what we have is all of RefSeq, all of antiviruses. There's a bunch of other stuff we started working on. Basically our approach is to build generic parsers that can take in different databases. Sorry, I keep touching something that makes me go backwards. And then we use the NCBI taxonomy as the glue that sticks all this together. So we then merge all the sequences together at the species level. And what I want to point out here is that we're not taking a single representative HIV sequence or a single representative influenza sequences. We're taking all the HIV sequences that are in the NT database currently, although you could expand that, and all the flu sequences. So essentially what we're getting is a cloud of sequences. And the reason for that is viruses are highly diverse. And one way to deal with that would be to try to extend your algorithms to tolerate a huge amount of diversity. Another one is just to expand the diversity of your reference database. So that's the cheap and easy way to do it. So that's what we're doing right now. Currently, our database is about 1.2 trillion nucleotides, boils down to about 26,000 species. That's what we're looking at. There's always a trade-off between trying to get as big of a data set as you can and not being overwhelmed by, shall we say, poorly characterized sequences. So how do we know if the pipeline works? We spent a lot of time building a synthetic data generator that was as realistic as possible, and we did that for two reasons. First, when we got actual mosquito sequences, the first thing we did, of course, was try a bunch of pipelines, and everyone gave us totally different results, and we had no idea how to believe which one. So we said, okay, well, let's generate synthetic data and see which one's right. The other thing is, I have a probabilistic background, and my general bias is that if I can describe how to generate data, then probably the best way to learn the model is to follow the generative model and try and learn that back. So I'm going to describe the way we generate data, and then in describing that, I'm actually describing the model that we use to learn back the model. All right, so basically we build a graph structure up front. So this part is actually not the learning part, but in order to quickly generate metagenomic data, we basically built a high-level programming language that says, okay, let's define some root. In this case, I'll use Anopheles gambiae, which is the best characterized mosquito. It's the malaria vector. And then we'll say, oops, with some probability, each mosquito will eat some host, could be any number of hosts, and if it eats a host, some number of the reads that you would have gotten from the mosquito will be reassigned to that host. Each of those hosts will, with some probability, be infected with something, and of course the mosquito itself may carry viruses or bacteria. You can try to assign viruses to hosts however you see fit. So here's what a synthetic data set might look like. Once you generate it, you basically get a whole series of things that are in it and absolute read counts, and you get the proportion. And so our task from the metagenomic standpoint is going to be to recreate these proportions. So we get a bunch of random reads, we don't know where they came from. Truth will be to come back and say that 89.2% of the reads came from Anopheles gambiae, and 10 to the minus 7, 2 times 10 to the minus 7th reads came from whatever that is. So how would you generate data from this? The first thing you would do is you would randomly sample over these organisms based off this probability distribution. So you have a probability that your read came from an organism. Once you know that the read came from an organism, the next thing you have to do is figure out the location. So there's there it is. There's the probability of a specific location within the organism. And then once you have the location, you actually have to generate the read. So the probability of a read given the location and the organism. So there's lots of tools to do the second part for us. So we're using NCBI's ART sampler. What's nice about that is that allows us to model the error distributions of different sequencing profiles. So we can basically say, well, what happens if we were to use high-seq versus my-seq versus PacBio or whatever. So we can very quickly generate data that way. But the more interesting thing, I think, is that this, as I said, suggests an actual learning strategy. So if you want to build the model, essentially what we want to do is learn this probability of the organism. So what we first need to do is define a way of, or specify what we mean by these different probabilities. So the first thing is we're going to have some function of read similarity. So BLAST will give you back basically an edit distance, and you need a function to turn that into a probability. So if there's 4 mismatches, what's the probability of seeing a sequence with 4 mismatches if you knew where it came from? You need the probability of a length. I think there's actually a lot of room for improvement here, but right now we're just assuming a uniform distribution, so it's 1 over the genome length, which is actually an important point, because effectively what this does is it will bias assignments toward short genomes. So if you get a read that equally matches a virus and human, it's more surprising that it came from the virus versus human, because the human has billions of bases. And then this organism, the probability distribution of the organism, that's what our learning problem is. So again, our goal is to learn this distribution, ideally to recover this, and in the mathematical sense what we're trying to do is maximize the likelihood function. So Will insisted that you guys wanted to actually understand the model, so I'm going to go into this a little bit. Forgive me if this is too much. I'll try to go through quickly. Basically what we're doing is a mixture model, right? So we can boil this down to a weight times a number, and our likelihood function for a specific read is going to be the probability of seeing the read given it came from an organism, integrated over all the organisms, and then if we assume all the reads are independent from each other, we simply multiply them, right? So that's simple enough. This lends itself to an EM algorithm, and the way we set that up, of course, is we say, well, if we knew where the read came from, if we have this latent variable z, which is 1 if the read came from that organism and 0 otherwise, that makes the likelihood function very simple. It's just a bunch of products, which means that when we do a log, we get a bunch of sums. And now we see this nice structure where basically what we want to do in the classic EM sense is we want to estimate the probability that a read came from an organism. And then once we've assigned reads to organisms, we want to optimize this total function with respect to the weights. So this leads us to an iterative algorithm. And if you want to talk about more of this later, I will. I'm happy to oblige, but I don't want to dwell too much on this. The key point here is that by doing this process of estimating read assignments to organisms and then optimizing the weights and going back and forth, what we're effectively doing is sharing information. So if you have a read that matches equally well to 1,000 different organisms, you can't a priori assign it very well. So the assignment will essentially be 1 over 1,000 of these 1,000 different organisms. But if one of those, say human, has a bunch of reads that map really well to that, then that accumulates evidence toward human over, say, chimpanzee, and it'll start pulling all the other read assignments over toward human. So I'll show you that in action in a moment. The other little detail is, what's this function? Like I said, this is just going to be a function of the edit distance, because that's something that BLAST programs give us really easily, is a read will map to a certain location, and it will give us an edit distance, so we just use essentially a binomial function to get that out. And if you want, you can learn the parameters for that, or you can set that. It doesn't really matter. Okay, so here's our overall pipeline. So we have some unknown cloud, source cloud, that led to our genetic material. We send it off to our sequencing machine. We get back, depending on what we run, 300 million reads, 30 billion nucleotides. We compare that to our reference of 1.2 trillion nucleotides. We essentially blast it with relaxed cutoff. So we want, basically what we're doing right now is we want every hit that's within 80% identity, which is pretty lax. So, you know, if you take a read from a human and blast it against RefSeq with 80% identity, you're basically going to get every vertebrate back. Not quite, but close. And then we're going to run it through the mixture model to resolve the ambiguities, and then we'll do some visualization. So I want to spend just one slide talking about BLAST. If you've ever used BLAST, you'll know it's impossibly slow, so we don't use BLAST. We're following SERPI's lead on this. So SERPI, a couple years ago, they published in Genome Research their pipeline, and they spent a lot of time comparing a bunch of different tools. So you may have heard of these. There's BLAST, there's BWA, Bowtie 2. And basically the point is that there's one algorithm that stood out as being way faster than all the other ones, so we're using that one. It's called SNAP. And what's convenient for us is this was actually developed at MSR in Berkeley. So the guys that developed this are down the hall, so they can help us modify it as we want, which is also very helpful. So this is the tool we're using. BLAST is what people know, so I often call it BLAST, so we're really using SNAP. All right, so let's look at what a synthetic experiment looks like, which will give us a sense for how this thing works. So here's the truth plot. So this is, let me spend just a minute describing this. So this was that table I showed you before, the anopheles and a couple things that it ate and viruses. And basically this is showing the proportion of reads at different taxonomic levels. So on the outside of species, then genus, family, et cetera, all the way up. And what we're showing here is that, in this case, roughly 90% of the reads went from anopheles, and then there was some proportion to Homo sapiens, et cetera, and then a tiny fraction went to viruses. The color is going to be how similar the reads are to their source. So green is 100% similar, red is 80%, because that was our threshold. And this is truth with 1% error, so it's all about 99% identical to the truth. So our goal is to reconstruct this. So I said the first step is going to be to snap these reads to everything. So if you do that and just count up the results, you get kind of a mess here. But you can sort of see the contours of the right results. So remember, Anopheles gambiae was our source mosquito, and we see hits to all the various Anopheles that are in RefSeq. The human blasts out to all the different mammals. There was human, mouse, and dog, I think, in there, maybe human, mouse, and bat. So that blasts out all these things. The bacteria go all over the place. The next step, so the reason for this, of course, is that, as I said, most reads map to many organisms. So the vast majority of these reads map to 2 to 10 organisms. Some of them are just crazy and map to thousands, and very few of them are unique. So you can actually try and look only at unique reads, and you'll roughly reconstruct it, although your proportions will be all out of whack. But you're really throwing away a lot of data, and that breaks down in many situations. All right, so the next step is to essentially weight these reads by their percent identity. So if we get back, say, a hit of a read to human of 99% identity and a hit to chimp of 98% identity, the human will get slightly more weight to this probabilistic thing that I described before. And so that starts cleaning things up a lot. So now we see more hits to Anopheles gambiae, but we're still getting a bunch of hits to other Anopheles. Same with the vertebrates, bacteria, et cetera. And then the last step is to do the mixture model, which really resolves all this, and we get back to truth. You can compare that to truth, and at least qualitatively, it looks pretty damn good. So here's the same thing if we zoom in on the vertebrates. So here's truth. This is what we're trying to get at. If we do snap, again, that just goes all over the place because we're so similar to all of the other mammals. Do the probabilistic weighting, and that helps a little bit. It gives us just enough hook. If we look in here at humans, it gives us just enough hook over the others so that the mixture model can really take off and resolve that back down to what it was. You can see that we're not getting everything here. We still have some extra things that are going to fish and unspecified chordata. These are small fractions. These are about 5 to 10 reads or so that couldn't really be resolved. Same sample, this is the viruses, so this is truth that we're looking for. If we just blast against everything, those viruses, even though viruses are highly variable from each other, many of those reads will still map to many other viruses, often in the same family, but they still spread out. Again, here's our weighting, and then here's the mixture model. We largely recover where we started. All right, so we can quantify this because we know where all these reads came from, so we can look at read level accuracy, the species, genus, family, order level, And by genus level, I simply mean we sum up the probability that a read is assigned to all the species within the genus, and then that's our genus probability and so on up the taxonomy. You can look at this at the organization level, so you can do an RFC plot, which is basically if you were to sort all of your genomes based off the number of reads that were assigned to each other, and then march down that list with your true positive rate versus false positive rate, and your goal is to get an area under the curve of 1. So that looks good. And again, you can do this at different taxonomic levels. All right, so that's great. When we get to a world where RefSeq contains all genomes in the world, then we're going to be all set, and when those are well curated. That's not terribly realistic, although if you can't do that, then you're in trouble, so it's good that we at least were able to do that. So the next step is to mask out organisms of the same species. So we'll sample from a species. Once we do that, we'll toss that out of the database and see where those reads go. Of course, our accuracy will be 0 on the species, but our goal then will be to have good accuracy on the genus level. We can then keep pushing that. We can mask out genus, family, order, and basically see at what point does the algorithm break down, and this is just not going to work. All right, so here's that same data set once we mask out species. So again, we've masked out Anopheles gambiae, and you'll see that all those reads map to other Anopheles, which is good. I actually think it's good that we didn't pick one. So the algorithm is basically says, well, it's an Anopheles, but I don't know which one, which is useful information. Same with the chordata. Now we're starting to get more of this garbage down here, although they're a pretty small number of reads. And here's viruses. So this is a little bit harder because viruses are so different, but here's chikungunya, and it's still mapping to an alpha virus, which is what chikungunya is. At low % identity, but we can still correctly identify the genus. And same with the coronaviruses, et cetera. Now we are starting to get some interesting things over here. If you look at the retrotranscribing viruses, there was nothing like that in our data set. So where did those come from? These are pretty interesting. These are all endogenous retroviruses, so these are in the RefSeq. So for example, we sampled from human, we masked out human, but we didn't mask out the human endogenous retrovirus, so we still got that back. Same with the murine leukemia viruses and whatnot. So that's actually pretty interesting. I think this is both encouraging that we're getting this. It's also useful as we get into real data, not just that we see a mouse, but that we see a murine retrovirus, or endogenous retrovirus is more evidence that we actually, in fact, are seeing a mouse in the mosquito. So again, we can look at the quality here. So at this level, genus accuracy was 95%. Now this is somewhat artificially low. We sampled from humans, and there is no other genus and homo that's in RefSeq. So by definition, our accuracy on the humans was 0. But at family level, we're pretty good, and same with these ROC plots. So we can keep going. Once we get down to a novel genus, I'd call it, our accuracy is 71%. Which is not as good as I'd like it. Family level, AUC is decent. Once we get down to novel families, we're really out there. Now, it's worth pointing out that even at the genus level, most of the reads are not aligning at all, so we're starting to work with fewer and fewer reads to try and estimate things. So some of this is the problem with the aligners. How do you get the aligners to basically back off and not have an 80% threshold, but a 70% threshold without blowing up computation and everything? Now, one thing that's interesting to note is that the family level performance varies widely across taxa. So there we go. So the vertebrates, we actually do quite well at the family level. Bacteria, we're miserable at. The arthropods are okay. Viruses are honestly better than I expected. And this, of course, depends on which viruses you sample from. Now, some of this is that bacteria are not well defined. The taxonomy is not well defined above genus level, so there's only so good you can do in bacteria. But there's actually a more interesting problem here, and that is contamination. And I'm going to put contamination in quotes because the definition of contamination is somewhat semantic. But here's an interesting example. So Wolbachia is an obligate intracellular parasite or symbiote, depending on the species and what you think about it. And basically it's a bacterium that infects most arthropods. At least there's a species of Wolbachia that is known to infect most insects, at least, and some nematodes even. And they actually live inside the cells of these mosquitoes. So when you go to sequence a mosquito, you have two problems. First, you're not pulling out a tissue, you're sequencing the whole thing. So you have its whole microbiome that you have to sort out. But even within the cells, you have the Wolbachia, and of course there's phages that are infecting the Wolbachia. So this becomes pretty hard from the sequencing standpoint. And in fact, when you look at a lot of what we're getting, we sample from Wolbachia, because that was a realistic sample for a mosquito, but then we masked out Wolbachia, and those reads had to go somewhere, so they basically get assigned to other insects that have Wolbachia in the RefSeq genome. And in fact, you can look at this even at the viruses, we get the same thing. So here we're actually getting the Wolbachia phage WO, which is infecting the Wolbachia, which was infecting the insect. And so once we masked all that out, we say, look, we found the Wolbachia phage, and that comes up as an error. So that's an error I can live with, but it actually becomes a problem with the real data that we're starting to see more and more is this contamination level. And you can do pretty systematic scans. So the system we have, I haven't really talked about the architecture, but it runs fast, so we can do our whole pipeline end to end in about 10 hours if things go well. This means we can also do systematic scans for contamination. So something we set up, it took about an hour, most of which was actually sampling the reads, was, okay, well, let's just sample randomly from every single virus and all of the nucleotide database, and then let's blast that, snap that against all of RefSeq, and just count how many reads for each virus go to each reference sequence. And here I'm looking at the most common contaminants within the bacteria. So these are bacteria and the number of RefSeq genomes that they are found in. So some of these jump out. Lambdaphage, or sorry, Phi-X174, if any of you guys do NGX sequencing, will recognize that as what's used to calibrate the Illumina sequencing machines. So that's found in, what is that, 250 some odd RefSeq genomes. Lambdaphage is obviously studied a lot, that's all over the place. Most of these are phages, but as you go down the list, you'll find a lot of other things. One of my favorite, if you look on the other side of this, is a salmon reference genome. It has a complete copy of an H1N1 virus in the RefSeq genome. It's got coronavirus, it's got part of a rotavirus, and it's got part of HIV in its genome. So either this was a very sick salmon and very interesting thing that we should be studying, or this is heavily contaminated either at the Illumina level, or I'm not sure what level this is. But this is something we're seeing a lot of, and this is causing all sorts of problems. Now, the good news is when you have viruses contaminating larger organisms, our bias, our genome length normalization basically fixes this. So I can show you some real data that are heavily contaminated and see this happening. But you also get contamination the other level. And we haven't looked at this systematically yet, but there's several papers in the literature that look at human contaminants that have been embedded in bacterial genomes because people are sequencing human all the time. And that's much more of a problem because now we'll get a read that maps both human and the bacteria, and we say, hey, it's matching this short little genome over here, so we'll suck it over there when that's not right. So if we have enough human reads, the mixture model will do the right thing, but if you're in a mosquito and most of that's been digested away and you're looking at only 1 million reads or so, then that becomes more of a problem. So one of the near-term things we're looking at is really doing a systematic scan of all bacteria, all viruses, and maybe all of human against all of RefSeq. Like I said, it's something we can do pretty quickly, so I think that's something interesting to explore to see what we can do with it. One simple thing is just mask out those problems. Another simple thing is to simply publish it and let people know this problem. But if you have any insights into contamination of these things, I'm really interested in how you guys deal with that and or how you find it. So let me pause for a minute and show you some real results, if this will let me. So where do I want to start here? Let me start, actually, since I just did the contamination, I'll show you some results that have contamination. So like I said, while this was motivated by the mosquitoes, our goal is for this to be broadly applicable. So here's a data set that some people contacted us, said they had a bunch of bird samples, so they were looking for avian influenza. They didn't know how to analyze it because there's no good reference. Could we just run it through our system? So we basically did a drag and drop and ran it. They've actually blinded us to what the species is, so I don't know if we're doing a good or bad job. My call on this would be that whatever the bird is, there's no reference up to the family level, but that is clearly a Passeriformes, I think is the first level at which we have a call. So assuming that's right, I think this is pretty cool that we can identify that. A bunch of these reads don't map anywhere. If you go to the viruses, which is what they're really worried about, what they're really interested in, the virus that jumps out here is Phi-X174, which means that their Illumina run was heavily contaminated. We also find some herpes viruses and other things. So that's kind of interesting. Another data set that we looked at, some people contacted us with ticks. They were looking for Lyme disease and other things like that. So they had a bunch of tick data. I'm not going to show you all of them. Some of them were wild-caught ticks, and other ones were negative controls. They were reared in the lab, so they were known not to carry any of these bugs. So this was an Ixodes ricinus. We actually map it to a little bit of both, which is, it's interesting when you get into these bugs because a lot of them, they're highly variable. Just the population genetics, even within a species, there's lots of debates over what's a subspecies versus what's a species. And the references tend to be lab strains, many of which were isolated decades ago. So they are somewhat diverged from what we actually see. And the other problem that I'll talk about in just a little bit is that we also have, these are very incomplete genomes. But in any case, we identify this as Ixodes. If you look over the bacteria now, which is what they were interested in, we see rickettsia, which they PCR confirmed. This candidatus, which is this metacoria, which is well known to be in ticks and whatnot, and some other things that were of some interest. Borrelia was the other thing they were specifically looking at. So that worked pretty well. You can also, they weren't interested in viruses, but we can see that there were some viral fragments in here. And one of the things that we are working on next is being able to actually add into the pipeline coverage maps and being able to do variant calls, so you can actually pull out the whole sequence. Because what I can't tell you right now is, was this 13,000 reads to the same region, which might indicate contamination, or are we actually covering the whole genome? So there's lots of tools that will do that, so we're in the process of just hooking those in right now. Some of the more interesting things, one, right? Yeah, so this is one that we just did recently. This was a positive control. So this was a kidney sample from somebody that had had a renal transplant, and they had PCR tested it for, now I'm going to forget the virus. There's a virus that causes this problem. It's polyoma virus, there it is. So they'd done NGS sequencing just as a proof of concept to see if this would work, and they sent it to us. I want to point this, I bring this up for a couple of reasons. So first, yeah, we can find humans, so that's good. And yes, we found the polyoma virus, and they sent us negative controls, and we didn't find it, so that was all great. What's kind of interesting, and what I want to point out a little bit just from the algorithmic standpoint, is that we did great on synthetic data in terms of resolving human, but we're still getting a bunch of other things. We're still getting pan-metagenomics in here, one of the chimp-related things, and a bunch of other mammals over here. And why are we doing this here? We're still looking into this, but really the root of the problem is that this is RNA-seq data, and all of our references are DNA. Right? So as soon as we get RNA-seq, the blasting process becomes much more challenging, because now we have splice junctions, we have all sorts of, we have poly-A signals and whatnot. So this is all messenger RNA specifically. And these become really hard to blast. So the problem is now, if BLAST or SNAP fails to get a map to Homo sapiens, it might still get a map to something else, but now we have a 0 in our probability matrix, and we can't possibly assign it to humans, so it has to go somewhere else. So this is one of the next steps when we talk about next steps in algorithmics. What we're not using is the taxonomy. So there's a bunch of taxonomic information. NCBI gives us a whole tree of life. It obviously has its issues, but it's at least a reasonable starting point. So you can start imagining trying to share information, right? So if you see a read that maps to a whole bunch of monkeys but doesn't map to human, it's actually reasonable to think that what you've actually done is hit a hole in that human genome, and it really should have mapped to human as well. So now you can start imagining extending the probabilistic model to say, okay, every read with some probability will have hit a hole, in which case we would observe a 0, but it really shouldn't be, so let's borrow the information from elsewhere. So that's one of the things we're really pushing on right now, to try and deal with this issue where you get incomplete matches. And the other, of course, is just to try and improve the SNAP and other BLAST-like algorithms to better deal with these splice junctions, although they're fundamentally a difficult problem. I thought I had in here a mosquito example. Maybe it's over here. Am I doing on time? I just can actually see the time up there. Oh, there it is. 4 more minutes. Okay, I'm wrapping up right now. Let me launch one more. So this was actual mosquitoes we caught in Grenada. This was, again, RNA-seq. This is total RNA ribosome depleted, although you still get a ton of ribosomes, which is challenging because those map everything. These were, in fact, Culex fasciitis, so we got the right species. Again, we have this hole in the genome problem. But this were 70 different mosquitoes that were wild-caught. They had not fed on anything, or at least they were not visibly fed. So it's good that we're not mapping this to any of the vertebrates. You can look at the viruses that they carried. And we actually are finding a couple viruses that are well known to be in the area. There's a Culex virus. There's this virus that, when we first got it, it was a really weak hit to other rhabdoviruses, and then the database was updated in January with this virus, which had just been described, isolated from Culex in the Caribbean, which is where we got these from. So we basically rediscovered that, although we didn't do any of the hard work of actually showing us what we discovered. So, you know, one of the things that, like I said, what we don't have is coverage maps. We sort of hacked this together here, but this is sort of the next step. There's all sorts of off-the-shelf programs. Basically, this is what you want to see. You want to be able to go back and map the locations, see that you're covering the whole genome, see where the variants are, pull down the sequences. So, like I said, there's lots of tools to do this. So this is what we're working on now to make that together. All right, let me wrap up here. Is that my talk? All right, so summary. So snapping against all RefSeq and NT is actually feasible. This is about 10 times bigger than what we've ever seen other people do. But snap is efficient with the cluster. and some biggish machines, we can get this to run in about 10 hours, especially for the DNA. The RNA-seq is harder because it maps to so many more things because the RNA is just more conserved. But we can get this to run pretty quick, and we think we can get this down a lot further. A lot of this is actually disk I/O problems that we can be a little more clever about how we're doing that. From the algorithmic side, so the other obvious thing is that the mixture models are highly effective at resolving ambiguities by sharing information across reads. So I think counting is a good idea. There's other approaches that do the mixture models, which is where we got the idea from. We approached it a little bit differently, and I think really focused on the scaling problem, trying to get this to scale up to really big problem sets. But this is really efficient. This whole thing takes about an hour, and 55 minutes of it is disk I/O. So the algorithmic part of it's actually very, very efficient. And there's obvious ways to make this more efficient. We're doing this with batch EM, which is memory intensive. But you can do stochastic gradient descent and all sorts of things that basically allow you to do it in real time as the data come in. As soon as we can get some more developers, we can work on that. So our goal, as I said, is to get to a point where we can do drag and drop metagenomics. I think there's obviously room to improve on here, but I think this is really feasible. I think it's a problem when we start trying to put in too much prior information. And when the prior information is good, that's a good thing. We want to use that. But I think for a lot of these applications, we really don't have any prior information. We really want to compare against everything that's known. So that's our goal. Next steps, as I said, really a principled approach to contamination and leveraging the taxonomic structure. These are actually linked. The first approach to contamination is to identify it, but there's some other more clever things you can do in terms of biasing the model to really pick genomes where you're covering the whole genome as opposed to concentrating all the reads in one spot. So we've got several interesting ideas to pursue there. Adding coverage maps, et cetera. Ultimately, our real goal is to port this all to a cloud service. That was the other thing we heard back from a lot of people, is that existing tools, even the best ones, are really a pain in the butt to use. So I think this is where a lot of cloud computing can be really helpful, is if we can just get it to work and provide an interface where you can do the drag and drop and we can do the compute. The trick is figuring out how to get the compute cost down to the point where that's beneficial for all involved. But I think we have a path forward for that. So let me wrap up there. I'll again bring up some people that we worked with. I focused again on the metagenomics. A lot of these people are doing the drone stuff, which I didn't talk about at all, and the robotics as well. But let me wrap up and take questions, and let's go get some beer and pizza. Yeah.
So you have some data where you're using DNA data and then RNA data. Is there a reason that you might use the DNA data or the RNA data from the mosquitoes, or this specific example?
The reason to use the RNA in the mosquitoes is we're looking for RNA viruses. The reason for DNA is it's a lot easier when you can use it. So if you're looking for the bacteria, it works a lot better. Again, the RNA causes a bunch of problems, but I think the problems that need to be solved. You know, one thing that we do that I glossed over is when we're building the databases, we grab all of RefSeq. We don't just grab the genomic data. Where available, we grab transcriptome data, so that helps some of that. But of course, that's sparse. Not many species actually have that. It's predicted for every species, but I'm not sure how much I trust predictions, especially when you move outside of well-characterized genes. And transcriptomes are diverse, so I don't know if there's such a thing as a transcriptome for a species.
What made you decide to use the EM algorithm over some other probabilistic model parameterization?
It's really easy. It's really easy. I mean, so I think, like I said, we started from the standpoint of how would we generate data, both for actually generating data for synthetic data, but also then that defines a model. And in this case, the model itself it just fits really well with the EM algorithm. The way it's specified is actually convex if we're not learning the mismatch rates, and it converges really rapidly. The biggest problem is we're loading the whole thing into memory. So for the DNA, you can get it to run on a small machine, and by small I mean it only has 128 gigs of RAM. If you do the RNA-seq where these things are matching to every vertebrate, then you suck up about 1 terabyte of RAM. So that's kind of limiting. Although the reality is that a machine with 1 terabyte of RAM only costs $40,000 or $50,000, and if you're spending a lot on sequencing, maybe you should buy a big compute to actually deal with it. But like I said, that's one of the reasons why we want to move this into the cloud. But the other thing is that it's just the easiest algorithm that we know of that we can just iterate and try different things with the models. But eventually, we will move that to more of a stochastic gradient descent, so that we can, you can imagine you have a cluster of all the blast to snap algorithms, and as those things are coming off, you can start integrating the data in real time rather than writing that on the disk, reading it all back into memory and iterating on it. it was just an ease of coding issue, and then once we have that right, we can do the more interesting stuff. Yeah, Richard?
With the trap, I wasn't sure whether you get one mosquito in there or you get a million. in each cell.
So yeah, so in the current iteration, each cell is one mosquito. There's 64 cells. 64 because the entomologist we worked with said 65 was a good day, and we're computer scientists, so we do powers of 2. You can imagine other designs. This was, V2 prototype. There's different use cases. So one of my favorite things I've learned about mosquito control is that one of the things that they do a lot to figure out if they have a mosquito problem is pay people to go out into fields and stick out their arm and count mosquitoes, which doesn't scale very well and has all sorts of problems. So, a simple thing to do is just turn off the closing the door and simply measure encounters. There's all sorts of designs that you can do with this, I think. And some of them would be to say, okay, well, if you want to capture 1,000 mosquitoes, then we need to rethink, rather than going to a cell, let's just suck them into a central holding tank. And the trade-off then is now you're losing the information that this particular mosquito was caught at this time under these conditions. So it's sort of a trade-off of what you want. Yeah.
I was wondering about how, so male mosquitoes don't bite, right?
Yeah.
So any viruses that you get in them are going to be passed down. And then if some of the diseases don't get passed down, then that's going to lead to a sort of overrepresentation in...
Yeah, so like I said, our goal is to move you from disbelief to doubt on the grandeur project. So one of the biggest weaknesses, there's an issue of prevalence, you might call it. So how many of these mosquitoes actually bit whatever carried it? How many of them actually have the virus that you could actually... there's a numbers game here.
Yeah.
Part of the point here is to say, okay, let's focus down so we're only capturing the things that we care about. So the first thing is, you can focus on Aedes, which bites human, or you can focus on Culex, which bites birds, depending on if you're looking for something like that. An open question is whether you can distinguish male from female mosquitoes. So as I understand it, it's not my area of expertise, the males are actually attracted to the bait as much. One of the guys on the team, his expertise is in what attracts mosquitoes, so a lot of what he's working on is how do you identify attractants that are very specific. The other problem is that most of the baits tend to attract hungry mosquitoes, and we want, full mosquitoes. So, that's an interesting question too, right? So once a mosquito eats, it massively increases its overall body weight. Does that change its wing beat frequency? That's never really been studied, so that's an interesting question. Even if you're not attracting many, if you can only capture the ones that are fed, because you do get ones that are fed in there. The ones that we've sequenced a couple of those, unfortunately they came back as garbage sequences full of adapters and everything, so we have to go back and do it again. But we are catching them, they're just at a low proportion of the overall sample. So that's one of the questions is, can you selectively capture them? Can you change the bait? Is it a fool's errand to try and do it at all? I don't know. Yeah.
Do you do any, collaboration with the guys over at RefSeq saying, hey, we found large amounts of Bi-X in this genome? Because I guess that's something that you're going to be coming across a lot with your EM algorithms. And go back and say, hey, this genome, in these spots is hitting kind of across the board.
So, so we have to solve this problem for our own purposes, and for that we are going to do a more systematic scan, like I said, basically tile all viruses, tile all bacteria, figure out where they are. It takes us on the cluster about, I think it was 4 minutes to run that scan once we have the sequences, so it's very efficient. So our thought was, quite honestly, we just discovered this issue pretty early, and I honestly don't know the literature for this very well. I don't know if there's tools out there that are already doing this. We've done a cursory review and found a couple of papers, but we need to really dig deeper in that. If it's a new thing, then what we'll probably do is stand up a service that's just a list of here's what's going on. We can monitor RefSeq and take every time you get something new, add it to the list. And what I'd love to do is find some contacts at RefSeq and see if they want to work with us on it. But if nothing else, we can only stand up a service that sort of points out which genomes are contaminated where. So I don't know. We got to kind of figure out if that's already been done, if we're just redoing this, or if there's really a need for this.
Showed that one genome coverage profile, showed good uniform coverage more or less. And with your issue of holes in the reference genome, those reach mismapping to other species. If you looked at the coverage of that species, that say, for example, that say wasn't present at all in reality, but it's all these mismapped reads that will pile up, and show non-uniform coverage.
Yeah.
How can you incorporate that information of like uniformity of coverage into your model?
Yeah, no, that's a great question. So I think there's two related but separate issues. One is the holes in the genome. The problem there is that you're getting 0 hits. The true source is here, and the related one is here. You're getting no match here, and you're getting a bunch of matches here. So you can imagine looking at it and saying, okay, well, the only ones we're assigning here are, in this problem region. That doesn't necessarily tell you should reassign them over here. So that's sort of a taxonomy sharing information problem. The other problem is, one way to look at contamination, again, is that if you have a bit of human that's been embedded in this microbe, then anytime you get human, you're going to get really intense hits in that one spot. And our current algorithm will say, well, that's great, that's a short genome, so let's focus there. But that's not correct, because it's all in that one spot. So we're exploring different ideas. One of the cool things that happens in machine learning sometimes is that you can have a generative model that generates the data. But one way to sort of put a check on that model is to say, okay, now that we've generated the model, conceptually, what if we were, or identified the model, conceptually, what if we were to generate data from that model? Does that actually fit our original data? So you can sort of go in both directions, sort of a bidirectional sampling. And then you can constrain the model to try and optimize both those models simultaneously. And what's cool about that is once you work that all out, it actually becomes a pretty simple optimization problem. Basically what you're saying is as I'm trying to fit the data, I'm adding in a constraint that the reads should cover uniformly a genome. That last part is the part that I'm struggling with most, because I don't want to cover it uniformly. If I'm doing RNA-seq, I know it's going to be spotty. But it's going to be spotty across the genome. So we've got to figure out how that works exactly, and if we need to modify some of those standard ways of doing that. Sort of there's a lot of bookkeeping, because now you have to keep track of locations. I think conceptually that's a really promising approach, but we've got to see if we can actually make that work.
You mentioned kind of early on that in some cases, you have sort of spotty reference, not that much reference.
Yeah.
Data, right? Like what? Just an estimate of how much payoff would you get by sort of making some yourself, right? Like you catch a bunch of mosquitoes, take some, sequence them, and then punch up your data that way. Yeah,
that's a really interesting question. And in fact, there's an argument to be said that in the context of premonition, we could just forget about RefSeq entirely and start collecting mosquitoes, and basically, as we get reads, basically build clusters, right? And then maybe try and analyze those more. And then there's something in between, which is, okay, we get this that looks kind of like Culex. There's a problem with Culex. We know it's not fully assembled. Now let's go to the effort and assemble it. We are adding a bunch of reads. I think that's a great approach. Right now, we're just simply limited by manpower. There's a lot of work to do those. But yeah, absolutely, we should be doing that. I would even say, if I'm just talking about cool research ideas, I think there's a huge, when you're talking about these things, there's reference mapping and then there's de novo assembly. And as far as I can tell, those are two totally different worlds, and I don't see why they should be. Because basically a reference is a set of pre-existing contigs, and if you're doing de novo assembly, you could match to an existing contig or a new one. And if you're matching to existing ones, you could start filling in those holes. So I think there's actually some really cool possibilities there, but it would be a pretty serious research project to do that. So if you know anybody interested in pursuing that. Yeah.
What's the half-life of a virus inside of a or a nucleic acid?
That's an excellent and critical question. And if it's arbovirus, it's a long time. So the ones that we found are arboviruses, right? They're replicating the mosquito. But yeah, this whole thing hinges on, can you actually isolate a non-arbovirus, non-mosquito-borne virus in the gut? So the good news is we're testing that starting on Monday. The bad news was it took us way too long to go through all the various approvals to get that done. But the plan is we're going to do a bunch of mosquitoes, taking human blood, spiking it with, I think we're using dengue and influenza because it's what we could get our hands on in short enough order, at known titers, and then feed a bunch of mosquitoes and then start killing a subset every hour for, I think we're going to do it for like 3 days, and then every day out to 25 days. And basically start off with PCR to see what's the decay in terms of what you do, qT-PCR or quantitative PCR, and see what you can detect with PCR, and then pick a few of those time points and go back and run the whole program. But yeah, that's a critical question. I wish I could answer that. It took us longer than we wanted to actually get all the right pieces in place for that. Yeah.
A sort of related question. You say you catch a hungry mosquito, right? What do you expect to be in that gut? I guess is kind of a related question. Right? Is there much residual stuff in there?
Yeah, it's unknown. That's why we're going all the way up to 25 days. I mean, we don't expect to see anything there, but that's sort of the full life cycle there. So we want to get the full range in that. So, the problem is I'm sure it depends on the virus in terms of how well encapsulated the RNA is. The DNA may be different. So we could do these experiments for a long time, but we're going to start on Monday and see where we go. I'll tell you, just to be honest, our initial seedling funding runs out in December, so we've got to get it in before the end of December, and then it's a process to figure out like what does the next phase look like, and that would be one of them.
Are you pushing all 64 together?
Yeah, that's also a good question. So the Grenada data that I showed you was, I think it was 70 mosquitoes. So those were actually mosquitoes we caught before the project started. We just went out and did it the old-fashioned way to just sort of see what this looks like. So those were 70 combined. What we just did that unfortunately came back is garbage sequences was just single mosquitoes. It's a cost-benefit trade-off, right? So I think ideally you'd sequence each mosquito by itself. But that's expensive, has a lot of data. At the same time, when we look at this project as a whole, I don't think we're, just from the sheer money side of it, we're in no position right now to be sequencing every track that we come back. But also, if there's a technology I'm willing to bet on in the next 5 or 10 years, it's sequencing costs coming down even further. So.
Yeah, for sure. Because you get these surveillance.
Yeah.
Applications and something like that. Pretty quickly if you do individual costs.
Yeah, absolutely. And that's one of the things we're talking about is sort of several public health agencies have asked, okay, well, can we just get a couple of these, leave the doors open, say put a cell card in here so that we can just leave it out there and have it just ping us with the information, and then we can come out and spray when it's an issue. So yeah, you can do that. And of course, from the classic surveillance standpoint too, if you're specifically interested in Zika or chikungunya, you should be capturing these things, pulling them, and doing PCR. Like, you're not going to beat that in terms of sensitivity. So somewhat of it depends on what you're looking for.
Yeah, but depending on how much DNA, I mean, you can be looking for people.
You can do what?
You can be looking for people. Like, specific groups of people.
Yeah.
So you can see applications going that way pretty cheaply, especially if it's drone-based, and especially you can imagine a kill in there with the dry ice so that, you don't have a half-life issue.
Yeah.
Pretty quickly.
Yeah, and in fact, you can imagine, so it's all, where does the technology go from here? So this thing's too heavy to go by a drone, especially if we don't have any bait in here. This can get a lot cheaper. This thing's made out of solid steel right now to make it super robust, but that gets small enough. The goal is to have this be delivered by drone. Once you're doing that, you can imagine preserving the mosquitoes on site. There's sequencing technology that's being developed that you can imagine even doing this here, although I'm not sure how much benefit there will be. At some point, you're always going to have the problem that the data need to get off here. I don't think we're going to get big enough compute on anything like this anytime soon, so at some point you're going to have to send the data somewhere else. But
Is there any kind of benefit to looking at male mosquitoes at all? Are you sexing them before?
Yeah, so I'm new to entomology, but it's interesting all the different ideas people get. So there's a lot of projects right now in terms of releasing genetically modified male mosquitoes, for example. So in that case, you might want to be capturing them to figure out, what's their prevalence in the population? Are they doing what they're supposed to be doing? So yeah, one of the open questions is, can you actually distinguish a male from a female based off wing beat? We don't really have any idea on that right now. But yeah, there are scenarios where you can want to do that. Right.
So I'm assuming in your blood samples that you're getting your genomic data from lymphocytes and lymphocytes.
Yeah.
So in that case, you think this might overrepresent lymphotropic viruses?
Yeah. So we're not. Yeah, that's one of the other issues, right? Obviously, at the very least, we can only find things that are in blood, maybe on skin, but probably not. We're certainly not going to get, a liver virus. So yeah, that's one of those questions: is what exactly can you look for here?
And I guess kind of maybe a related question: how much viremia does a virus have to cause to show up in bloodmeal and then show up in your sample?
Yep. All good questions. I mean, this is what we're trying to do: the blood feeding experience.
Any idea of how sensitive this needs to be yet?
Yeah, that's what we want to do the blood feeding experiments before. So people do blood feeding experiments, but the focus in the literature has always been trying to determine whether the virus can replicate in the mosquito. So they haven't phrased the question in this way, and they haven't specifically done viruses that don't replicate in mosquitoes and titer them. So our initial run just for cost and complexity issues, it's just going to be a single titer of the virus. But if we can get funding, I absolutely want to titer down the virus within the actual blood meal, because that's a huge question. And, we can do some back-of-the-envelope questions in terms of, or estimates in terms of, like if you have a blood sample with, 10 to the 5 copies of HIV in it, and you do total RNA-seq, like what proportion of the reads are HIV versus human, and then you can try and compare that, okay, once you feed a mosquito, what proportions of the reads are human versus mosquito, and you can try and estimate it that way. But yeah, that's a big question. And one of the big technological things from the molecular biology side that I think is an open question in virus discovery in general is how do you specifically amplify virus without being specific to certain types of virus? Because if we can do that, then that makes the problem a lot simpler. And there's various strategies in terms of size particle filtering, trying to target conserved regions and pull them down. And we've been exploring some of those. We haven't had a lot of luck getting those to work very well, but we've only had a year, and I think there's a lot of work to do in that space. But yeah, I think that's a huge open question for the field as a whole, because in all these metagenomic studies, the first thing you do is throw out 90% of your data, and that's kind of dumb. Yeah.
I think you're going to collect a lot of data for this technology here. Do you plan on putting this open source data set?
Yeah. So that's we're trying to figure out what the right model is for that. So there's a lot of ways you can imagine doing this. I mean, so for things like the trap, we're not really interested at all in becoming a trap manufacturing company, I can tell you that much. So the plan is to open source the hardware design on that, and then focus. There's a lot that we're discussing. I can't tell you all of it, but I can tell you that Microsoft is making a big bet on cloud computing in general. So I think we can make a big case internally, which is that getting people to do computing in the cloud is a good thing to begin with. So in fact, a lot of this needs to be in the cloud. Some of the questions that I'd actually be interested in your feedback on some of this stuff. So we talk about a cloud-based metagenomics thing. If we have a drag and drop metagenomics, how much would you be willing to pay for that? In the short run, we can probably front all those costs in terms of grants to researchers, but at some point, the compute is really expensive. And so even if we do it at cost for compute, how much are people willing to pay for that? And I don't really know the answer to that. People pay for the sequencing. Are you willing to pay the same amount for the compute if it's good? Are you willing to pay half of that? And what's sort of the model for that? My personal approach, and whether I can convince everybody else to do this, is that I think we need to make as much of it as open as possible and as free as possible, whether it's through internal or external grants, at least initially, to understand what's working and what's not working. And then we need to figure out a way to make it sustainable, and we'll figure that out down the road. But that's either it's not going to work, in which case it doesn't matter if it's sustainable, or if it's going to work, then we need to show that it works up front. So, but I don't know if I can convince everybody of that. That would be my personal hope.
Is there a remote or doesn't reflect in whether the research model published?
No, so we're. Sabrina can attest to this. I have a hard time publishing things until I feel like everything is done, so I tend to work on things for a long time. So we're trying to figure out what the publishing strategy should be. I think I'm hoping we're going to write this up soon. We may pause and focus on the contamination issue first, if that's the lower hanging fruit. Some of these algorithmic things on the mixture model, I'd love to get it all done and then publish it as here's a final solution, but it might make sense to publish things along the way. One of the hard things that is always a struggle, I think, with research is that writing papers takes a lot of time. It's very important, but the question is, when do we pause and do that versus when we get pushing this? So I'm hoping that we can start that process in the next couple months. Sabrina will tell you that even if I start writing a paper, it may take a couple of years before I actually do it. But I promise not to take a couple of years. Cool. Well, thank you, guys.
So we have pizza and beer just down the hallway, so we'll move there now.